Okay, now let us move on to the next question. Which of the following antibodies constitute the largest proportion of the total antibodies present? Yes, students, I want to tell you about this, something called as GAMD, right? So what is GAMD? These are the different type of immunoglobulin. What is immunoglobulin? Immunoglobulin andre ಏನು? Just ಹೇಳಿದೆ ಅಲ್ಲ? Antibodies. Yes, students. Now antibodies 5 types ಅದು ಇರುತ್ತೆ, correct? The first type is IgG, IgA, IgM, IgD, IgE. How will you remember? GAMD. GAMD andre ella 5 types of immunoglobulins irutte. Correct alla? Okay. Now out of that, which is the one which is present in the largest proportion? And yes, students, the option here is IgG, because actually it comprises of 75% of the total antibodies present in the blood. And the rest one, if you see, again, the importance about IgA is what, you know, it is actually giving you a immunity when we are very, very young. That is the infant condition, IgA, present in the colostrum of mother's milk. The first milk of the lactating mother will be comprising of IgA, and IgE plays a very important role in allergic responses. Okay, so let us move on to the next question. So again, a NEET question which was been asked: which one of the following immunoglobulin does constitute the largest percentage in human milk? IgM, IgA, IgD, IgE. Just now ನಾನು ಹೇಳಿದೆ ಅಲ್ಲ, lactating mothers ನಲ್ಲಿ first milk ನಲ್ಲಿ yellowish fluid, that is a colostrum ಇರುತ್ತೆ, correct? ಆ colostrum actually present ಇರ್ತದೆ, ಇದು IgA, which is rich in that, and that's why it has been suggested that breastfeeding for the infants is very, very essential during the initial weeks after the delivery, because the child is given that antibodies to the kid, so I mean infant, so that it can fight or defend the pathogens. Okay, so that is a very important role played by IgA present in the colostrum of the milk. Okay, so far we have discussed some of the very important MCQs, right? Now we are going to talk about the different types of the acquired immunity. Yes, before ಸ್ವಲ್ಪ ನನಗೆ ಹೇಳ್ತೀರಾ innate and acquired difference ಏನು actually? Yes, innate immunity ಅಂದ್ರೆ from the birth, inborn, right? And acquired andre ಏನು? During the lifetime we develop it, when we encounter different pathogens in our life, so we slowly develop that acquired immunity. Slowly andre there are different ways how we acquire that immunity. So let us see what are the different types of acquired immunity, and here we go. So acquired immunity, as you know very well, ಇದು ನಾವು slowly develop ಮಾಡ್ತೀವಿ, ನನ್ನ ಬಾಡಿ slowly ಕಲಿತದೆ ಇದು immunity develop ಮಾಡಲಿಕ್ಕೆ. ಕಲಿಲಿಕ್ಕೆ ತುಂಬ ways ಇದೆ. Let us now learn what are the different ways. First of all, you will find in acquired there will be something called as active immunity. Something called as the active immunity. So let us now talk what is this active immunity. Yes, I am talking about this active immunity. Second one is the passive immunity. Right? So first of all, you will have two types. One is the active acquired immunity. Then comes is the passive acquired immunity. Now, what is the difference in active and passive immunity? I can give you some examples. Okay. For example, you know, we encountered some various infections, you know, like the sore eyes. Sore eyes ಗೊತ್ತಾ ನಿಮಗೆ? ಐಸ್ ಇರಿಟೇಷನ್, ಕಂಜಕ್ಟಿವಿಟೀಸ್ ಹೇಳ್ತೀವಿ, ಕರೆಕ್ಟ್? ಸೊ ಕಂಜಕ್ಟಿವಿಟೀಸ್ ನಿಮ್ಮ ಲೈಫ್ಗೆ ತುಂಬ ಟೈಮ್ ಬಂದಿದೆ, only 1 or 2 ಟೈಮ್ ಬಂದಿದೆ ಆ್ಯಕ್ಚುಲಿ. ಎಲ್ಲವರಿಗೆ ಕಂಜಕ್ಟಿವಿಟೀಸ್ ಆಗ್ಬೋದು, ಕರೆಕ್ಟ್ ಅಲ್ಲ? 1 time. ಸೊ ಹ್ಯಾವ್ ಯು ಅಂಡರ್ಸ್ಟುಡ್ ದ ಡಿಫ್ರೆನ್ಸ್? ಫಸ್ಟ್ ಟೈಮ್ ನಮಗೆ ಕಂಜಕ್ಟಿವಿಟೀಸ್ ಆಗಿದ್ದು, ದ್ಯಾಟ್ ಟೈಮ್ಗೆ ನಿಮಗೆ ಐ ಪೇನ್ ತುಂಬ ಸಿವಿಯರ್ ಇತ್ತು, ರೈಟ್? ಆಮೇಲೆ ಅದು ಟ್ರೀಟ್ ಆಗಲಿಕ್ಕೆ ಸ್ವಲ್ಪ ಜಾಸ್ತಿ ಡೇಸ್ ತಗೊಂಡಿತ್ತು, ರೈಟ್? How it slowly then decrease the days after first exposure, some years later you must have again developed that conjunctivitis, right? But that time, if you see, the irritation or the swelling in the eyes wouldn't have been so so aggressive like the way it was in the first time. That means slowly your body tried to develop that immunity, and yes, now it can defend the pathogen which can cause the sore eyes problem to you. Let us see what is that now. So first type what we are going to learn here is active immunity. Immunity you developed after being exposed to an infection. Okay, to an infection means it can be a natural infection, the way like I said for the sore eyes, automatically it happened to you and your body recognized it and slowly tried to defend the pathogen. Or from getting vaccine. Getting vaccine is not natural, that means it is going to be artificial. I hope things are clear to you now. So active immunity, if it is natural, meaning what? How will you acquire natural active immunity? Yes, that means if the body encounters the organism, that is a pathogen, I mean to say the pathogen naturally, okay, that time the body knows, okay, this is a new organism or the pathogen which is trying to infect me, and now that will be the first exposure, right? The first time you get that disease. That time the disease will be defended, the body will fight back, but the time taken for recovery will be more, right students? And that is how your body now learns to produce antibodies towards that pathogen. So that is a natural training given to your body by the real organism, correct? So that will be the, when your body encounters a natural infection, the immunity acquired by your body will be active natural immunities. That means you are going to make antibodies with the help of the natural infection, correct? Similarly, now what do you mean by artificial? Artificial andre yenu? Now nimge vaccine kodtivi. Vaccine andre yenu students? Yes, vaccine andre, it can be the dead or weakened pathogen. Dead or weakened or preparations of the organism. Matlab it can be the toxins of the organism also. I mean the toxins of the pathogen also, which can be injected in the form of vaccine. So any of the form will be called as the form of vaccine. It can be the dead pathogen, or it can be the weakened attenuated pathogen, or it can be some of the chemical substance of the pathogen in a weakened form. So any of them can act as a vaccine, and when you inject this vaccine onto your body, so body now understands, okay, this is something, a type of organism, yes, a pathogen, and now slowly your body is getting trained in an artificial manner. So indirectly you can say vaccination is training your body to produce the antibodies towards that specific organism or the pathogen, right? So now the body knows, okay, for example, we are injecting you with vaccination A. So vaccine A will have a pathogen A. So okay, now your body is injected with vaccine A. So this will be the antigen. So now the body knows, okay, this is a type of antigen. In future it can attack me. Okay, let me now try to produce antibody towards A. So when the next time when really that antigen A infects your body, so that time the memory cells which are there, the type of the memory cells produced by the adaptive immunity will encounter this antigen and produce the same type of antibody and then confer you with that immunity, and that is going to be artificial active immunity. Hope things are clear to you. I gave you with the examples also. So example here could be a disease like chicken pox also. Chicken pox only once in lifetime we encountered, right? Chicken pox ಬೇರೆ ಯಾರಿಗೆ second time third time ಆಗಲಿಲ್ಲ. ಆಗಿದ್ರೆ ಆಲ್ಸೋ ಅದು ಲೆಸ್ ಸಿವಿಯರ್ ಆಗುತ್ತೆ. ಫಸ್ಟ್ ಟೈಮ್ ಅಂದರೆ ತುಂಬ ಸಿವಿಯರ್ ಆಗುತ್ತೆ. ಸೆಕೆಂಡ್ ಟೈಮ್ ಥರ್ಡ್ ಟೈಮ್ ಚಿಕನ್ ಪಾಕ್ಸ್ ಆಗೋದಿಲ್ಲ. ಯಾಕೆ ಅಂದರೆ ಯುವರ್ ಬಾಡಿ ಆಲ್ರೆಡಿ ಅದು ಇಮ್ಯುನಿಟಿ ಡೆವಲಪ್ ಮಾಡಿದ್ದಾರೆ. ಅಂದರೆ ಬಾಡಿಗೆ ಅದು ಮೆಮರಿ ಇದೆ, ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ಗೆ ಮೆಮರಿ ಫಾರ್ಮ್ ಆಗಿದೆ ಕಿ ಇದು ಟೈಪ್ ಪ್ಯಾಥೋಜನ್ ನಮಗೆ ಬಂದಿದ್ರೆ ನಾನು ಇದು ಟೈಪ್ ಆಂಟಿಬಾಡಿ ಕ್ರಿಯೇಟ್ ಮಾಡಿ ಫೈಟ್ ಮಾಡಬೇಕು, ಗೊತ್ತಾಯ್ತಾ? ಸೊ ದಟ್ ಈಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ನ್ಯಾಚುರಲ್ active immunity. Second, with the help of vaccine ತಗೊಳ್ಳಿದ್ರೆ ಏನು ಡೆವಲಪ್ ಆಗುತ್ತೆ ಅಂತ that would be artificial immunity. That is artificial active immunity. Now can you think of what is passive immunity? Yeah, passive, that means something with respect to ready-made antibodies. Passive andre in one simple word you can think of ready-made antibodies. Ready-made andre again two types irutte. This one is a very simple antibodies, that is a natural passive form wherein the mother to child, okay, the newborn baby, the infant, okay, which is just a few days old, from the day one after the delivery of the baby requires immunity. Remember student, adu chikka maguge infant ge actually immunity ಏನು ಇರುವುದಿಲ್ಲ. acquired immunity develop ಆಗಲಿಕ್ಕೆ ಟೈಮ್ ಆಗುತ್ತೆ. ಅದಕ್ಕೆ mother's milk one type of antibody ಇರುತ್ತೆ. ಏನದು? IgA, colostrum ನಲ್ಲಿ, right? So ಅದು IgA definitely ಅದು mother to child transfer ಆಗುತ್ತೆ through the milk, and that is how the baby is going to get the immunity. Yes, that is what ready-made antibodies, plus it is going to be ready-made but natural form. That's why it is called natural passive immunity. Things clear? Mother to child, again natural passive immunity. Ready-made antibodies but in the natural form. ಇದಕ್ಕೆ ಯಾವ ಎಕ್ಸಾಂಪಲ್ ಕೊಡಬಹುದು? Passive artificial ಅಂದ್ರೆ sometimes you must have encountered a dog bite. Okay, rabies dog bite agbodu. So there are we get scared if there is a dog bite. There are chances that rabies infection can occur, right? For that reason, after a dog bite, we are suggested to take antibodies. You mean to say we are supposed to take rabies injection. So that rabies injection definitely would be what? Preformed antibodies. So that preformed antibodies which are in ready-made form, but now it is in the artificial form. Since we are injecting it, that's why it is going to be called as what? Artificial passive immunity. Things clear? So these are some of the types of the active and the passive acquired immunity with some examples of natural and the artificial. Now let us talk about some of the examples here with the MCQs. Okay, look at this now. As I said, active immunity, you will be having the example of natural and artificial. Natural meaning the real natural infection, the pathogen enters and trains your body and really the body learns to make the antibodies. So next time if there is a second attack, the body knows to produce which type of antibodies, antigens are to be killed with that type of the antibody. So always remember students, for every type of antigen there is every specific type of antibody. Like the lock and key, a specific lock has got a specific key. Similar way, for a specific antigen there is a specific antibody present. So that antigen antibody interaction will take place here, giving you the actual active natural immunity. Now go to the artificial one, that is with the help of the vaccination, because we are injecting from external source. That is what the vaccines will create that immunity. Next year, the maternal antibodies from mother to child through colostrum, that is IgA, and then the monoclonal antibodies, the way I said for the snake bite, even snake bite can be example for this. Okay. So here we give with the example what is the basic principle of immunization and vaccination. Okay. Basically, there are two different terms here, immunization and vaccination. So what is vaccination? The process, or you can say the act of giving vaccine to the individual will be called as vaccination. So what is immunization? Yes. So when you give a vaccine to an individual, the vaccine will be taken by the organism and slowly the individual, okay, the human will try to recognize this as an antigen, and then against that antigen, antibody will be produced, right? So all the cascade of the mechanism will take place. In turn, you can say for towards that vaccine, a typical antibodies are being trained to be formed. So when that entire process, the response for the vaccine, the body is showing back to the vaccine process will be called as what? The immunization, right? So here we go with the option. What is the basic principle of immunization and vaccination? It is based on the number of B and T lymphocytes. It is based on the property of memory of the immune system. It is based on antigen-antibody interactions, based upon the number of white blood cells. Try to strike out the one which is not required. So you can see it is not at all that connected with the options. Even here, it is nothing to do with the antigen-antibody interaction, right? So what is left out is A and B. Based on the, is it the number or is it on the property of memory? Think of that, students. Naan just helide alla. The best property or the characteristic of acquired immunity ಏನಂದ್ರೆ memory. Acquired immunity ಗೆ actually memory ಇದು ಕಾನ್ಸೆಪ್ಟ್ ಗೊತ್ತಿದೆ. ಅಂದರೆ ಅವರಿಗೆ ಸರಿಯಾಗಿ ನೆನಪಿರ್ತದೆ ಯಾವ ಟೈಪ್ ಪ್ಯಾಥೋಜನ್ ನಮಗೆ ಇನ್ಫೆಕ್ಟ್ ಮಾಡಿದ್ದು. ಸೊ ಸೇಮ್ ಟೈಪ್ ಆಫ್ ಪ್ಯಾಥೋಜನ್ ಸೆಕೆಂಡ್ ಟೈಮ್ ಇನ್ಫೆಕ್ಟ್ ಮಾಡಿದ್ರೆ ಏನು ಮಾಡಬೇಕು, ಯಾವ ಟೈಪ್ ಆಂಟಿಬಾಡೀಸ್ ಮಾಡಬೇಕು. ದ್ಯಾಟ್ ಪ್ರಾಪರ್ಟಿ ಆಫ್ ದಿ acquired immunity plays a very, very important role here, student. So our option would be, it is based on the concept of the property of memory of the immune system. So this is what the MCQs we try to cover on the vaccination as well. The answer here would be B. And now we move on to again a similar type of question. Vaccine against polio virus is the best example of, what is it? Auto-immunization, passive immunization, active immunization, simple immunization. When you talk about the word active, that means it is vaccines. Vaccines come under what? active, and that is active. When we talk about active, it is artificial or natural. It is going to be artificial active. So the answer here would be C, active immunization. And that also which type? Artificial active immunity, adaptive active immunity. Okay. So here one more question related to that. Which of the following statement is wrong regarding vaccination and immunization? Immunization is a phenomenon of increasing specific antibody production. Vaccination is a process of inoculation of harmless antigenic material into healthy person. A single vaccination gives adequate immunity and lasts lifelong. Immunization is carried out through vaccination and injection of anti-serum. So according to you, which one is a wrong statement, student? Can you think of that? So I give you a just 10-second break and we'll come back and just let me know the answer, right? Okay. ನಾವು ನಿಮಗೆ ಸ್ವಲ್ಪ ಬ್ರೇಕ್ ಟೈಮ್ ಕೊಟ್ಟಿದ್ದೀವಿ ಸ್ಟೂಡೆಂಟ್. ಆನ್ಸರ್ ಸಿಗ್ತಾ ನಿಮಗೆ? ಓಕೆ, ಹೇಳಿ ನನಗೆ ಆನ್ಸರ್. I repeat the question. Which of the following statement is wrong regarding vaccination and immunization, right? Look at the first option. Immunization is a phenomenon of increasing specific antibody production. Correct ಇದೆಯಲ್ಲ? When you take vaccination, immunization, ಇದು actually development ಆಗ್ತದೆ antibody, right? Vaccination is a process of inoculation of harmless, correct? Harmless antigenic. That means, what was that harmless antigenic meaning? Yes, as I said, vaccine andre adralli ಏನು ಇರುತ್ತೆ actually? Vaccine andre ಅದಕ್ಕೆ ಏನು ಇರ್ತದೆ? The attenuated organism, or you can say the organism is weakened, okay, weakened, or it can be dead organism, right? So it can be weakened or dead antigenic matter, or it can be a form of the organism, a small part of the organism, which can be also called as antigenic, having that antigenic property. So not necessary. Vaccine can be either made up of a dead organism or the dead pathogen, or it can be or attenuated pathogen, or it can be any part of the pathogen which can trigger the stimulus of acting like an antigen, or it can be any toxin produced by the pathogen, right? So any of them can be used as a vaccine. So this comes under this category, harmless antigenic material into healthy person. That is also correct. Single vaccination gives adequate immunity. Look at the word. They are telling ಒಂದು ವ್ಯಾಕ್ಸಿನ್ ತಗೊಳ್ದ್ರೆ ಸಾಕಾ? ಇಲ್ಲ. ಕೊರೋನಾ ಟೈಮ್ಗೆ ನೀವು ನೋಡಿದ್ದೆ ಅಲ್ಲ, ನಿಮಗೆ ಒಂದು ಪ್ರಾಪರ್ ಡೋಸ್ ಸಿಗ್ತದೆ, ಆಮೇಲೆ ಸ್ವಲ್ಪ ತಿಂಗಳ ನಂತರ ಬೂಸ್ಟರ್ ಡೋಸ್ ಇರುತ್ತೆ, ಗೊತ್ತಾಯ್ತಾ? ಬೂಸ್ಟರ್ ಡೋಸ್ ಏನು ಮಾಡುತ್ತೆ? ಆಂಪ್ಲಿಫೈ ಮಾಡ್ತದೆ, ಸ್ವಲ್ಪ ನಿಮಗೆ ಬಫರಿಂಗ್ ಮಾಡ್ತದೆ, ಬೇರೆ ಆಂಟಿಬಾಡೀಸ್ ವಾಪಸ್ ಸೇಮ್ ಟೈಪ್ ಆಂಟಿಬಾಡೀಸ್ ಜಾಸ್ತಿ ಮಾಡಲಿಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ. ದಟ್ಸ್ ವೈ ಫಸ್ಟ್ ವ್ಯಾಕ್ಸಿನೇಷನ್ ವಾಟ್ ಯು ಗೆಟ್, ಡೋಂಟ್ ಥಿಂಕ್ ಇಟ್ ಈಸ್ ಲಾಸ್ಟಿಂಗ್ ಲೈಫ್ ಲಾಂಗ್. It doesn't last for the lifelong. You have to take a booster dose, right? So last answer again, immunization is carried through vaccination and injection of anti-serum. Again, that is correct. The option here, which is the option C, is not the correct one, because single vaccination doesn't give you a lifelong immunity. You need something called as booster dose of the same vaccine at specific intervals. That is very, very, very essential. Even ಮಕ್ಕಳಿಗೆ ನೋಡಿ polio vaccine ಕೊಡ್ತೀವಿ ಅಲ್ಲ, ತುಂಬಾ ಟೈಮ್ ಅವರಿಗೆ booster dose ಕೊಡ್ತೀವಿ ನಾನು. ಯಾಕೆ ಅಂದರೆ so that they should keep developing that immunity and that there should be continued immunity be available. That's what one single dose cannot give you a lifelong immunity, right? Okay, so we move to the next concept. See, a NEET question which was been asked in 2016. Anti-venom. Anti-venom andre which type of immunity? Anti-venom against snake bites, right? Anti-venom andre passive immunity. Passive andre adaptive or acquired passive immunity. Can you tell me whether it is going to be artificial or natural passive? When we say injections given externally, that means it is going to be artificial. So I'm talking about artificial passive immunity, right? Okay. Antivenom injection contain preformed antibodies. Preformed antibodies are nothing but the ready-made antibodies. Ready-made antibodies. While polio drops are administered into the body content. Now polio drops, what are these made up of? Yes, think about the answer. Polio drops will be either attenuated pathogens, activated pathogen, gamma globulin, harvested antibodies. See, all the four options ನಿಮಗೆ ಇದು ಸ್ವಲ್ಪ not connected ಕಾಣುತ್ತೆ, ಕರೆಕ್ಟ್ ಅಲ್ಲ? ಸ್ವಲ್ಪ ನೋಡಿ, ಇದು ಬೇರೆ ಆಪ್ಷನ್ನಲ್ಲಿ. ಈಸ್ ಇಟ್ ಆಕ್ಟಿವೇಟೆಡ್ ಪ್ಯಾಥೋಜನ್? ನೋ, ಆಕ್ಟಿವೇಟೆಡ್ ಪ್ಯಾಥೋಜನ್ ಇರುವುದಿಲ್ಲ. ವ್ಯಾಕ್ಸಿನ್ ಅಂದರೆ ವೀಕಂಡ್ ಆರ್ ಅಟೆನ್ಯೂಟೆಡ್ ಪ್ಯಾಥೋಜನ್ ಆರ್ ಡೆಡ್ ಪ್ಯಾಥೋಜನ್, ರೈಟ್? ಸೊ ದ ಆನ್ಸರ್ ಹಿಯರ್ ಇಸ್ ಆಪ್ಷನ್ ಎ, ಕರೆಕ್ಟ್? Yes, students. Now we are going to discuss about the next disease, and that is AIDS. Let us talk the MCQs related to the disease AIDS. The first MCQ, the human immunodeficiency virus is. First option, an unenveloped RNA genome containing retrovirus. Next, an enveloped RNA genome containing retrovirus, an enveloped DNA genome containing the retrovirus, and an enveloped RNA genome containing reovirus. So looking at all the three options, one thing is sure that this is not connected with them. It's not a reovirus. In fact, HIV virus is a retrovirus. Let us see how does it actually multiply. Okay, this is how. Now imagine now this is one retrovirus, right? Now this retrovirus, that is the HIV virus, is found to have the RNA genome, and this RNA genome is having a coat, that is a protein coat, which is a very strongest asset of the viral virus here. Okay, so viral protein coat is going to protect the viral RNA, right? Now remember one thing, students, the virus has got its RNA but not its DNA. So now let us see how it is going to convert its RNA into the DNA, right? So once the viral, that is the RNA containing the viral coat protein, that is the RNA virus, attaches to the host cell. Okay, imagine now this is one host cell, and now you can see here the host cell will be attached in such a way that slowly the viral RNA is introduced into the host cell. Now there is one special enzyme called as reverse transcriptase. This reverse transcriptase, by the process of reverse transcription, will convert this RNA into the DNA, right? So now the virus has got converted its RNA into the DNA form. Slowly this viral DNA will try to get incorporated into the host genome. So this is the host DNA, right? And now the viral DNA, which just now got converted from RNA to DNA, will get incorporated into the host DNA, and slowly now this will behave as if it's their own DNA, and then it will try to transcribe and translate all the proteins. In fact, it is going to transcribe and translate all the proteins as per the message present in the viral RNA via the DNA. So if you see, the new viral RNA will be produced, and then there will be packaging. So they will be packed in the viral protein coat, right? So these are the new viruses which are produced, and yes, these new viruses are again going to contain the RNA in it packed, right? And slowly they will rupture the host cell, and now these are ready to go and infect the other cells. Mainly remember one thing, students, in the case of HIV disease, that is, you can see AIDS, in the disease AIDS, actually the main problem lies because the main target is been done to the T helper cells. Yes, students, you know we studied about the immune cells. There were B cells and T cells, right? So amongst the T cells, there were different type of T cells, like the cytotoxic T cells, the helper T cells, the suppressor T cells, and all. So if you see, the helper T cells are the one which were actually helping the B cells to trigger the entire mechanism of immunity. But here in AIDS, it is found that the viruses actually go and infect or destroy the T helper cells. So in turn, you can understand, once the T helper cells are damaged or attacked by the viruses, that is HIV virus, definitely you can understand the immunity of the person drops down, because the helper cells are getting decreased, and in fact, if you see, the person's immunity goes down in such a level that the individual actually cannot fight with the normal infections or the smaller infections also. So overall, if you see, the entire immunity is degraded because of the lacking or decrease in the number of T helper cells, and slowly the person now gets, becomes a host for many infections. So it starts with tuberculosis, etc., etc. So this is how overall immunity is being brought down, and the person loses its entire immunity, and slowly as I said, in the case of the AIDS disease, the death is inevitable, but it can be little prolonged, but the death is inevitable, right? So this is what actually happens in the case of the HIV virus life cycle. So now try to understand the options here. So the options here are, now you are knowing the answer very well. Is it either A or B? Of course, it is not having the DNA, but it has got an RNA. And I said this is not the right option here because it is reovirus. We are talking about the retrovirus, right? And the answer here is going to be option B. As I said, it has got a RNA genome containing the retrovirus with a protein coat surrounding itself, right? Okay. Next question. The agents causing cancer are known as. So we moved on to the next concept, that is next disease, that is cancer. And the first question here, the agents which causes the cancer are known as oncogenes, carcinogens, antibodies, cannabinoids. Students, antibodies are something which are connected with the immune cell, that is B cells produce antibodies, so it is not connected here. Cannabinoids are basically the drugs. Again, it is not connected here. Now, what matters is the two genes here. Oncogenes. Oncogenes are the cancer-causing gene. Onco stands for cancer. Remember one thing, students. There are certain genes which are called as oncogenes if they are triggered or stimulated by some external substances which can trigger and convert them into cancerous cells. So those genes will be called as what? The oncogenes. And what is this left out? Carcinogen amongst the option. Yes, students, there are various agents which can actually damage the DNA, or you can say alter the DNA in such a way that the entire cell can be made cancerous. So those agents, okay, which will lead to the transformation of the normal cells into cancerous neoplastic cells. Neoplastic meaning the cells which will show uncontrolled cell division and form masses of cell, that is tissue masses, may be induced by either physical, chemical or biological agents. And yes, these agents are actually called as what? The carcinogens. Some of the examples you can give is the UV radiations or also the smoking, etc. Right? So this was some of the questions related to cancer, and the answer here is B. Next, we move on to this question, a very important and new concept about the cancer. Which of the following is false regarding neoplasm? Let us now understand what is this neoplasm. As I said, in the case of the cancer, it so happens that a normal cell, if you see the journey of a normal cell, look at this. This is how the normal cell appears, right? And this is how the status of the cancer cell. If you see properly, the normal cell will have a large cytoplasm, whereas the cancerous cell will have a little cytoplasm. In fact, the nucleus size will be more in such a way that the cytoplasm decreases, and here in the normal cell, it will have a definite small size of the nucleus, but which is not in the case of the cancerous cell. Second is, it will have a single nucleus. You will find here they will have multiple nuclei. Also, the chromatin network will be dense here, fine, but here you will find coarse chromatin network. So somewhere you can understand the DNA content, okay, or the genetic content will be damaged or altered in such a way that they will lead to abnormal cell division. So this is how it appears. In the case of normal, we had studied, right? One cell divides to form two daughter cells, two to form four, four to form eight, etc. That is what? Controlled division, right? And you know, in case of the cells which are with damaged DNA, look at the status of the cells dividing here. One cell will divide into two abruptly. It can lead to 8 or multiples, and it can, abrupt cell division which can lead to multiple mass of cells forming a abnormal tissue growth. And you know what is a very important aspect found here? Generally, when the cells divide, you know, there is something called as contact inhibition. Now, imagine this is one framework, okay, and contact inhibition is shown by the normal cell. Meaning what? Contact inhibition meaning, suppose now there are 5 cells which have divided, and they will adhere to themselves in such a way that it will not allow the extra cells to grow. So there is something called as contact inhibition. But in the case of the abnormal cell, that is the DNA damaged cancerous cell, the contact inhibition property is lost. So that's the reason it is found that there will be overclumping of the cells, abnormal dividing, and there is masses of the cells which is called as tumor being formed. And remember students, tumor can be of two types, that is benign and malignant tumor. Out of the benign and malignant is found to be one of the dangerous one, because The malignant tumor has got the capacity to spread to various parts of the body. So if you see this tumor which is found, it will definitely have the chances that it will spread and it will go and lodge somewhere, and there only it will form a new tumor. So that process, or you can say that concept of spreading of the tumor, will be called as metastasis. It is called as what? Metastasis. So metastasis is a concept where there is spreading of the tumor, and that's the reason why in the cancer many tumor will be formed. That is how we can understand the main difference lies because the cancerous cell do not have the property of controlled cell division, which is lacking in the case of the cancerous cell, right? Okay, so here was the question, and the options here with respect to neoplasm is, which is false regarding neoplasm. It is abnormal tissue caused by abnormal proliferation of the cells, cannot spread to other body organs, and responsible for the formation of the tumor. So here we are asking which is false regarding the neoplasm, and yes, the answer is neoplasm. Basically it cannot spread to other part of the body organs is actually wrong. In fact, it spreads, and that is a concept, that spreading of the metastasis is a property, what I said, shown by the malignant tumor. So neoplasm is a new abnormal tissue capable of continued growth, okay? The formation of the tumor, the disrupting of the normal cells, they are caused by abnormal proliferation. So main reason lies here because of the abnormal proliferation, and they may or may not be localized, okay? So answer here is option C, cannot spread to other body organs, okay? Now, the last part of the chapter is about the drug and alcohol abuse, okay? And here is one question regarding that. Cocaine, there are certain drugs like cocaine, opioids, cannabinoids, etc., okay? So you need to know the plant from which we are deriving it. Remember one thing, student, these drugs basically which are found are actually drugs generally used for medicinal properties. Suppose if a person has got pain after surgery, you know, the patient is administered with painkiller so that he can overcome the pain. But if the same drug is been taken in an overdose form, it becomes drug abuse, which is really not good for the health. And in turn, if there is continued drug abuse, definitely there will be deterioration of the health of the person because it directly affects the nervous system. Okay, so here are some examples of the MCQs here. Cocaine is obtained from Erythroxylum coca, Papaver somniferum, Datura, Atropa, belladonna. And the answer here is it is developed actually from Erythroxylum coca. And yes, this drug which is there is generally used as a local anesthetic and also used as a vasoconstrictor. Vasoconstrictor meaning narrowing of the blood vessels. So the blood vessels can be narrowed of the blood vessels. That is a property shown by cocaine, and it's a powerful stimulant. This is a plant name and which is native to South America. So this is actually sniffed in powder form or taken as a drink. So this is one commonest example of the drug, that is cocaine. And here we have the answer here, that is, as I said, Erythroxylum coca. And the next question here, LSD. LSD stands for lysergic acid diethylamide, okay? So it's basically taken from a fungus, that is what the option A, and it is basically the Claviceps purpurea, which is actually the fungus from which we extract the drug, that is LSD. So these were some of the MCQs based on the entire chapter, students, and I hope now you have understood the very important aspects of the chapter called as human health and disease. Now, there is very, very interesting news for you all, students, and here it goes. Diksha is going to organize a CET crash course, right? And yes, this is just going to start after your board exams, okay? And it will be for the duration of 45 days. And yes, students, this course will be for the residential as well as the non-residential students also. And you know what the benefit of the course? You can solve the questions in a very quick and a very efficient manner. In case we are in fact, we are going to give you a lot of practice, and which will help you to remember the concepts at the fingertip. And yes, the very important part of the CET is that within the time limit you are supposed to solve that, which will be trained here at Diksha. So see to it that you join this crash course and avail the benefits too. Look at some of the toppers so far we had here. Ronak Birangi with 2nd rank, Pallavi with 3rd rank, 5th rank Siddhartha, and 6th rank Vaishnav. So if you want to be amongst the best colleges, right, and you want to be your name to be present here like this, please do join. And now, why are you waiting and what are you waiting for? Just start with the registration. Call at this number, that is 1800-102-4109. And yes, there is a link given below which will give you all the instructions for that. And by then, students, we'll be back with another chapter, important questions of that chapter. By then, students, signing off for today. Dr. Smita Narayan Karthik. Thank you.